Hi, welcome everyone to the Cancer Genomics Cloud webinar for April 2024. I'm very excited to present our speaker today, Dr. Basil Bakir. Dr. Bakir is a clinical fellow in medical oncology at New York Presbyterian, Columbia University Irving Medical Center. He completed his medical scientist training program at the Perelman School of Medicine at the University of Pennsylvania, and then went on to an internal medicine residency at the Johns Hopkins Hospital. And after starting his medical oncology fellowship, he joined the laboratory of Dr. Ken Olive, and he focuses clinically on gastrointestinal cancers. Dr. Bakir is with us today because he is the 2023 recipient of the PANCAN, the Pancreatic Cancer Action Network, Cancer Informatics and Data Science Research Fellowship. So this is his talk presenting the work that he has done with the Pancreatic Cancer Action Network. With no further ado, please take it away, Dr. Bakir.
So much for that introduction, and thank you for giving me the opportunity of speaking to everyone today. So I'm going to talk to you today about a project that I'm working on in Dr. Ken Olive's lab at Columbia with the Seven Bridges Genomics group regarding biomarkers for chemotherapy sensitivity in pancreatic cancer. So my agenda today is I'm going to present, just some brief background on pancreatic cancer, then talk mostly about the conceptual backbones for a computational tool called master regulator analysis, followed by telling you what we're doing right now with the database and our very early starting analyses. So pancreatic cancer. So pancreatic cancer is a challenging disease, and it's challenging for a couple of reasons. So first of all, pancreatic cancer presents with very vague presenting symptoms. Patients often, when they come into their primary care doctor, who end up having pancreatic cancer eventually, come in and tell their primary care doctor, doc, I'm feeling full. I'm having this vague abdominal pain. Most of these symptoms are pretty nonspecific. Sometimes you have something more profound, like jaundice, yellowing of the skin, because there's a pancreas mass blocking the bile ducts and bile juices. Or you can have extreme weight loss if the pancreas is really not making enough of the enzymes it needs to make to digest food. But often the presenting symptoms are vague, and there's really a significant amount of time between the presenting symptom or when the patient first notices it to their full workup and diagnosis. And so vague initial symptoms plus the absence of any easy screening for this cancer means patients often present late, and more than half of pancreas cancer patients present when they're already stage 4, metastatic. So we're dealing with a cancer that has pretty vague symptoms and no easy screening, a bad combination for patients. And this leads to poor outcomes. So the latest data from the NCI shows a 5-year survival rate of 12.8% across all stages. So this is even early stage. Now, 12.8% is, of course, not good enough. And as a field, we have to do better. But it's maybe worth emphasizing that crossing 10% was actually a huge milestone for those of us in pancreas field. For years upon years, 10%, what we were in the single digits, and we just in the past few years have crossed 10%. Here you can see that survival is really directly linked to what stage you present in. This is again data from the National Cancer Institute SEER database, showing that patients with localized disease have a 5-year survival of about 44%. And then when you're dealing with patients who are distant, stage 4 disease, their 5-year survival is 3%. And part of these poor outcomes are also driven by the challenging biology. So immunotherapy has revolutionized many cancers, melanoma, for example. But pancreas cancer is virtually entirely refractory to immunotherapy. It doesn't have any of the biological characteristics that make cancers susceptible to immunotherapy, including a high tumor mutational burden or what we call MMR deficiency. In other words, the cancer has all the proteins needed to fix its DNA. And we know that not being able to fix your DNA is one of the things that sensitizes cancers to immunotherapy. The second thing is pancreatic cancer is pretty bland from a genomic point of view in terms of oncogenic drivers and targetable mutations. Virtually all of pancreatic cancer, 95%, has a KRAS mutation. There's P53 at high levels in SMAD4, but neither P53 or SMAD4 are targetable with drugs. And KRAS is generally not targetable. There are some very exciting drugs coming down the pipeline, but they're still in clinical trials. And so when you put all of this together, you put together vague presenting symptoms, the absence of easy screening, challenging biology, that's what really drives these poor outcomes. And that's what we're trying to work on. So let me talk a bit more about the clinical need. So there are basically two chemotherapy backbones that are used in pancreatic cancer. One is called FOLFIRINOX. FOLFIRINOX is what we call triplet chemotherapy, in that patients get 3 different chemotherapy drugs. FOLFIRINOX is just an acronym that includes, the sort of starting letters of each drug. So the F stands for 5-fluorouracil, the I stands for an irinotecan, and the O stands for an oxaliplatin. These are nucleoside analogs that incorporate into DNA or other mechanisms of damaging DNA. The other backbone is gemcitabine nab-paclitaxel, which again functions. Gemcitabine is what we call, again, a nucleoside analog that works via something called masked chain termination. Basically, the cancer cell incorrectly incorporates the gemcitabine metabolized version into DNA. Then there's a normal nucleoside placed against it, but then you're done. There's no more DNA synthesis. And nab-paclitaxel is again a drug that blocks cell division by inhibiting microtubules. Basically, in almost all settings for pancreas cancer, before surgery, after surgery, or for patients who never go to surgery, these are fundamentally the two backbones we use. There are very rare instances where we know any kind of genetic data that drives us towards one or the other. One notable exception is with BRCA, a gene that sort of has wide resonance because of its close association with breast cancer, which we do find in pancreatic cancer. For patients who do have a BRCA mutation, roughly 5 to 10% of pancreas cancer, you know that FOLFIRINOX is generally going to work better than gemcitabine nab-paclitaxel. But for everyone else, you really don't have a great way of knowing which regimen to use. And because many of our patients unfortunately present late stage and unfortunately maybe don't have a lot of time, giving the patient the quote-unquote incorrect regimen results in loss of valuable time. And lastly, and maybe this is potentially one of the most significant things, FOLFIRINOX has a much worse side effect profile than gemcitabine nab-paclitaxel overall. And if we can find something that helps us not give FOLFIRINOX to patients who aren't going to benefit, this is going to lead to a much better quality of life. So the purpose of this project is to use a technique called master regulator analysis to discover biomarkers of sensitivity or resistance to either 5-fluorouracil, FOLFIRINOX, or gemcitabine-based regimens in pancreas cancer. So what I'm going to do now is I'm going to walk through 3 different concepts I want to introduce. And I'm going to present these concepts on a qualitative level, not a quantitative level. I am not a computational biologist, but I'm very lucky to be at an institution that has incredible computational biologists in our systems biology department. So I'm going to kind of give you the perspective of how someone who's more in the clinic thinks about this. So the first concept I want to introduce is the concept of the regulon. So let's imagine you have some arbitrary protein X, and you know that protein X has a certain effect on the transcriptional profile of a cell. So protein X upregulates mRNAs A through D, and that's represented by the green color. And protein X not just upregulates them, but has stronger and weaker connections to each mRNA. That's represented by the thickness of the arrow. So protein X, we would say, positively regulates expression of target mRNA A or B or C or whatever. Now, let's look at mRNAs E through H. Protein X suppresses those mRNAs. It decreases them. So protein X negatively regulates expression of these mRNAs, and again, represented by the thickness of the arrows, has a different effect. So the regulon of protein X is the tissue-specific map of how protein X first affects the expression of any given mRNA and how strong that interaction is. So the regulon, to put it another way, is the relationship between protein activity and transcriptional profiles. So that's the regulon of any given protein. Now, let's make this slightly more complicated. And instead of just looking at a protein, let's look at a cell. And I'm going to present the most simple cell ever. This box represents a cell that has two proteins in it. Let's look first at protein X. Protein X upregulates A and B and decreases C and D. And protein Y decreases A and B and increases C and D. So X and Y have the exact opposite regulons in this cell. So what does that mean? Well, all of these regulons together, regulon of protein X and regulon of protein Y, represent the tissue-specific transcriptional network in that cell. And so let's imagine let's now imagine you have a cell and it has very little of A and B mRNA, very little of it. Well, you can probably infer that protein X isn't very active because protein X, if it was active, would mean you'd have a lot of A and B. And if you have a lot of C and D, you can infer that protein Y is very active because there's a lot of what protein Y upregulates. So fundamentally, this is about using transcriptional networks to infer protein activity. So I want to just take a moment here and emphasize why this is kind of a big deal. When I was first starting this project and trying to wrap my head around it, I had to really sit down and understand, why is inferring protein activity from transcriptional networks so important and such a sort of revolutionary thing that the systems biology group, Dr. Adria Califano at Columbia did? So the first, partially, is just technical reasons. You can't do a Western blot for every protein in a sample. Protein can be assayed high throughput through reverse protein phase arrays, but generally this is more expensive and harder to do than high throughput sequencing of DNA or RNA. So with the first reason why this is a big deal is because it lets you understand more about a hard to assay macromolecule proteins using an easier to assay macromolecule RNA. The second is for biological reasons. So work done at Columbia in the systems biology group has shown that there is a remarkably small set of proteins that are responsible for executing a cellular phenotype associated with cancer. That whatever that cancer phenotype is really depends on a few signaling nodes that execute it. And these proteins are termed master regulators. Now, you might very reasonably say, why is that a big deal? Of course, we know that certain proteins execute a cellular phenotype because we know cancer has mutations, and those mutated proteins are driving the cancer. So here's one of the things I want to emphasize. A master regulator protein is not a mutated protein. A master regulator protein can be an entirely wild type protein, not even increased in amount in the cell that executes that phenotype. And so master regulators allow you to decouple mutated oncogenes and drivers and lets you understand how are potentially non-mutated proteins driving cellular phenotype. So this is now the third concept, that of sort of master regulators. So let's say you have a cell that has oncogenic drivers and non-oncogenic drivers in Y through Z. Well, what master regulator analysis has shown is that these diverse driver mutations, their signaling and their effects are catalyzed into a very specific tumor master regulator module. And these master regulator modules are not necessarily what's mutated. They're often not mutated. And then this master regulator module is what regulates the cell's transcriptional profile. So to phrase it differently, if you look at driver mutations or the transcriptional profile alone, you're going to miss a key layer of regulation, namely the master regulator. And again, I'll just emphasize this, MR proteins, master regulators, do not by definition have to be mutated, and so you'd miss them if you were doing DNA sequencing. So this is all, for me as a medical oncology fellow, very rarefied stuff when I was learning about it and sort of reading what the computational biologists were thinking about. What I want to emphasize is that this is not theoretical. There is a massive amount of experimental evidence that master regulators actually exist, actually drive phenotypes, and are executing the cancer phenotype in many, many different cancers, including glioblastoma, prostate cancer, and neuroendocrine tumors. And I'm just going to very briefly touch on these three just to show you an example. So the first tumor type that was ever interrogated through master regulator analysis was glioblastoma, a form of brain cancer. And they looked at a prognostically poor subtype of glioblastoma and showed that there was a 6-protein master regulator module that regulated 3/4 of the cell's transcriptional profile. And going back to this idea that master regulator proteins are not necessarily mutated or differentially expressed, these 6 proteins were not themselves differentially expressed on a transcriptional level. So if you were relying just on RNA sequencing to look at what drives the cellular state, you'd have missed this. The second is prostate cancer. So this is one of my favorite examples because the GU oncology group, the prostate biology group at Columbia with Dr. Califano in systems biology, did a cross-species analysis of human prostate cancer biopsies and 4 separate mouse models of prostate cancer. And they found that that almost half of the master regulators in each mouse model were conserved in human prostate cancer, which tells you that there's something really elemental about these master regulators as drivers of the cancer, in this case, prostate cancer, because you found them across 4 separate mouse models and then validated them in human samples. The third example is one of my, I think one of the most powerful. And what they did here is they took pancreatic neuroendocrine tumors. They asked, what drugs can I give that block the master regulators in neuroendocrine tumors? And do I see a regression of those tumors in various models, including a subcutaneous mouse model? And they did. They predicted, based on master regulator, what would be the driver of PNET, pancreatic neuroendocrine tumors, did a drug screen to find what blocks it, and then validated it. In fact, master regulator analysis is so well validated that it forms the basis of a non-experimental, clinically approved assay that can be ordered by any physician in at least New York State. You have to get state approval for each of these assays, and it's at least been approved in New York, and I believe in California. And so, if you're if you're an oncologist in New York State, you can just go to Columbia University's Department of Pathology, order the test, and that's it. It's not a clinical trial. It's completely standard. Now, you might have trouble getting insurance to pay for the drug, but that's a different issue because that's kind of a separate issue. But the assay itself is completely validated. So far, I've tried to present a couple of different ideas. The idea of the regulon, which is the idea that any given protein has a different effect on the transcriptional profile of a cell. I've presented the idea of the transcriptional network, which is the idea that if you take all the regulons of a cell together, you get a map of how those proteins interact. And then the idea of master regulators, which is the idea that there are specific signaling nodes that catalyze the genetic drivers in any given tumor. So how do we do this analysis? How do we take a patient sample and get a master regulator profile? And this is something called VIPER analysis. VIPER analysis stands for Virtual Inference of Protein Activity by Enriched Regulons. And what you do is you take any given cell's transcriptional profile. So here we have a cell that has very little of A and B and a lot of C and D. You put it through the VIPER algorithm, which essentially operationalizes regulons and transcriptional network analysis. And then based on VIPER analysis, you can infer which proteins are more or less active based on the transcriptional profile. So in this case, with a cell that has very little of A and B and a lot of C and D, you would infer that the proteins that are most active are the ones that upregulate C and D, protein Y. And so, what I'm doing right now, and I'll sort of, so there are a couple of different ways to go back to the starting genesis of this project. I told you at the start of this project that I was interested in using master regulator analysis to understand how different patients respond to chemotherapy in pancreatic cancer. So one way of doing this would be a prospective clinical trial where you enroll patients, randomize them to one or the other regimen, see how they react, how they do on the regimen, and have RNA sequencing of those biopsy specimens, etc. This is, of course, a huge undertaking to do a trial like that, very expensive, very time-consuming. But we have a pretty wonderful tool, which I'll describe in a moment. So what I'm doing right now, or what you would do in a project like this, is you take RNA-seq data from 5 FU responders or gemcitabine responders, put it through VIPER analysis, and then get a master regulator profile of 5 FU responders and gemcitabine responders. And then you'd go back to human tissue to validate with immunohistochemistry and various other assays that the proteins that you think are driving this response are actually driving it. You'd start in human tissue, you'd then start doing xenograft models or organoid models where you knock in or knock out the proteins, that kind of thing. So the big question is, where am I going to get this RNA-seq data? I mentioned that one thing is you could do a prospective trial, which is a giant undertaking beyond this, the scope of what I can do. Luckily for us, this data set already exists. So the Pancreatic Cancer Action Network has a database called SPARC, which has annotated molecular and clinical data from 650 pancreas cancer patients in the Know Your Tumor program. This includes various things like chemotherapy treatments, survival data, and tumor markers, and most importantly, or one of the two most crucial factors, the RNA sequencing data itself on the tumors. And because this data already, this project would have been inconceivable for me without this Spark database existing. And so what I'm doing right now, and we're sort of at this stage in the project, of making the cohorts and putting them into the pipeline to start. I don't yet have any putative drivers of resistance or response, but I'm hoping to have those really over the summer, early fall, and then start validating in human tissue and eventually potentially in organoids, mouse models, and things like this. I want to sort of lastly end on what are the challenges when working with a data set like this. So you first have to generate paired cohorts of tissue that are matched on several clinical and pathologic characteristics. When did the patient receive their chemotherapy? Was it before or after the biopsy or surgery? The chemotherapy itself is probably going to change the master regulator profile. So I'm trying to avoid comparing patients who got chemo before their biopsy and patients who got it after. Assessment of tumor response. I've been telling you that I want a cohort of 5 of responders and non-responders to one regimen and responders and non-responders to a different regimen. So how am I going to define that? And some of this is going to be a little heterogeneous. Things that are going to come into play are tumor markers. Some of the patients in the Spark database have tumor markers that, go up or go down and can be used as surrogates of response. Some of those patients are going to have potentially some data on their imaging responses over time. Did the tumor shrink or not? And some of them, I'm just going to be able to know how long they were on any given treatment. So one of the challenges with putting together cohorts is being kind of very careful in defining what a responder and non-responder is and making sure sort of the groups are balanced and make sense in that regard. Excuse me. The third challenge when working with a data set like this is, patients often undergo dose adjustments and changes in regimen. I can tell you just in my experience as a fellow, these regimens are not benign. FOLFIRINOX, for example, the oxaliplatin, the OX component of it, causes really bad nerve side effects or really bad neuropathy. And for some patients, it can be so bad that you have to make significant dose adjustments or just, in some instances, remove it entirely for a duration of time. Irinotecan, the IR component, can have really significant GI side effects. And so patients, you can say they're on FOLFIRINOX or gem/nab-paclitaxel, but when you delve into the weeds, what you're really seeing is a patient who's getting half-dose oxaliplatin or was off oxaliplatin for a really long time. And so one of the things that I'm having to account for when making these cohorts of patients that we want to put into Viper is making sure that the doses that they've been getting are actually significant enough to be biologically relevant. If a patient is listed as receiving FOLFIRINOX, but only got oxaliplatin for one time and then was off it, is that really the same regimen? Can I include that patient with that group? And so the heterogeneity of treatment is a challenge when making these cohorts. And then I think one of the most interesting biological challenges when making these cohorts is related to tumor heterogeneity. So pancreas cancer is, when you look at pancreas cancer under a microscope, huge amounts of the tissue are not actually cancer cells, but stromal components. They're fibroblasts, they're immune infiltrates. They're not tumor cells. The RNA-seq data in the Spark database is whole bulk sequencing. So those transcriptional profiles represent fibroblasts, macrophages, endothelium. And in some cases, the tumor component, which the Spark database has, can be as low as 10 or 20% of the tissue that was sequenced. And so you can imagine if I'm working under the assumption that I'm interrogating a regulon of a pancreatic cancer cell, and my RNA profile is 80% non-pancreatic cancer RNA, that's going to require some thoughtful analysis and balancing between the cohorts, and our computational biologists are helping us with this. So, pancreas cancer has very few clinically useful biomarkers for chemotherapy sensitivity. A protein's regulon represents the relationship between protein activity and RNA levels. Our systems biology group has shown that the cancer phenotype is executed by a remarkably small number of master regulator proteins. Viper is the name of the algorithm that allows you to go from sort of the RNA sequencing profile to a list of candidate master regulators. And the purpose of this project is to apply Viper analysis to the annotated molecular clinical database in the Pancreatic Cancer Action Network Spark database, so that we can find biomarkers to pair patients more effectively with chemotherapy. I'd like to acknowledge Professor Ken Olive in the Olive Lab, my mentor, Dr. Andrea Califano, who was the person who pioneered master regulator analysis for the past 2 decades at Columbia. The Pancreatic Cancer Action Network for their data science fellowship, and access to the Spark database, and the Seven Bridges Genomics group, especially Kauthar, who's been helping me with this project. And then more broadly, the Columbia University Tissue Bank team and Dr. Gipshush will be important for this project when we start trying to validate in human tissue what we think the regulators are. I ended a bit early, and thank you so much for everyone's attention.
Thank you so much for that wonderful presentation. We do have a few questions from the chat, and then that'll also give other folks time. I'll voice these questions, and then other folks, if you also have questions, go ahead and use the Zoom reactions to raise your hand, and I'll let you voice them after these. So our first question for you was, are these master regulators of embryonic origin? And I wondered what you had to say about that.
Oh, that's such an interesting question. I actually don't know. The sort of last Dr. Califano's group published a 2013 Cell paper where they did really comprehensive. They presented sort of this model of, I believe it was 17 master regulator modules across numerous cancer types. I don't know if any of the proteins that they in that list were embryonic origin, but that's a great question. It would potentially make sense that they are, but I don't know.
Yes, I agree with you. It would potentially make sense. I know that in prostate cancer, one of the HOX proteins is there's a variant that's implicated, and so I'm sure that a lot of master regulators are that are probably active during embryonic development as well, because they're involved in directing cell phenotypes.
Oh, thank you. Yes, you're absolutely right. Yeah, with HOX and prostate, definitely. Yep.
Yes. And so another question we had was, as mentioned in one of your slides, the driver mutations are upstream or downstream of the master regulators plays a significant role. Viper works on transcriptomics data. Are there any tools that can utilize the genomic aberrations, mutations, copy number variants, or epigenetic modifications with Viper analysis to do master regulator analysis?
Again, that's a great question. I will ask my computational biology colleagues, but I don't know. I think they do have more sort of in the Dr. Califano group's GitHub. They have dozens of tools that are part of this toolkit. I believe some of them can kind of take it to that next level. It's not currently part of our sort of initial pipeline of what we're planning to do, but yes, I think those tools do exist.
Excellent. Thank you. And one final question, and this is maybe more of a conceptual question. Will master regulators reinforce the paradigm of cancer stem cells? I think this is someone who's thinking about, cancer stem cells.
That's a great question. I need to. I don't know that we have any experimental evidence that. So I guess the idea of cancer stem cells is in and of itself, I guess, a bit controversial. I won't get into that. I myself don't have a clear opinion of it. But to the extent that to the extent that we sort of want to accept that framework, I don't think there's any experimental evidence yet to reinforce it. I think it would be a really interesting experiment, for example, to see if, you take a cell line, you knock out or you upregulate, you reintroduce a master regulator, you force overexpression or knock it down, do any, putative cancer stem cell markers increase or decrease. Those would be, I think, really interesting experiments. But I know there's no experimental evidence yet for it, I believe, linking the two.
All right. I actually have a question of my own. I actually have a couple of questions. This is just a very clear and compelling talk. And one of the things that I found myself thinking about is identifying the master regulators that are implicated in pancreatic cancer. Sounds extremely interesting and informative as basic science. What are in terms of translational research, how does this end up translating to treatments for pancreatic cancer? How does this end up giving us either biomarkers or targets?
Absolutely. So let's say that this project succeeds in identifying a master regulator that's really tightly linked to, let's say, sensitivity to gemcitabine. So we do our pipeline, we find this unexpected protein that's very linked to gemcitabine sensitivity. We validate it in tissue, we validate it in subcutaneous models and xenograft models. Well, then you could imagine writing a protocol and trying to come up with ways where, you take the patient's initial biopsy, you do RNA sequencing on it, you do master regulator analysis on it, and you see, does that patient have high expression or not? I want to clarify, not expression. No. Yeah, exactly. Is that master regulator enriched in that patient's specimen? If it is, then that suggests you should use a gemcitabine regimen. Now, the challenge is the lag time, right? If a patient comes to you in clinic, needs chemotherapy, then do you have a well enough developed pipeline where you can run the RNA sequencing fast enough, do the master regulator fast enough so that you're not waiting weeks and weeks for the data? But long term, I think, the point you're raising is how would this be operationalized in the clinic? And I think this is kind of what we imagine if we're able to find exciting candidates, how it would work. That you would do RNA sequencing as a matter of course on a patient biopsy, do MR, and then pair the patient with the chemo based on that.
All right, that makes perfect sense. That's beautiful, and very, very hopeful for folks with pancreatic cancer, but also folks who are sometimes worried they may have pancreatic cancer in the future. It's a scary and debilitating disease. Do we have any other questions from attendees? All right, well, seeing no other questions, I'm going to also offer a comment rather than a question. One of the things that's really fascinating to me about master regulator analysis is and I think this came up also with our earlier question about embryonic regulators, is how much cancer is sort of a cellular developmental kind of process in so many ways, that the regulators that are implicated aren't mutated. They're just doing their jobs, and they've been told to do their job in a pathological way. And it reminds me a lot of some of the interesting stuff in evolutionary developmental biology about where like novel phenotypes come from, that, you know, these regulators are still just doing the same jobs, but they're being redeployed in new ways. I just think this is fascinating work, and I wanted to thank you for doing it.
Thank you so much for that invitation. And I just want to again thank the Pancreatic Cancer Action Network and the Spark database, which has been incredibly helpful and supportive. So thank you to them.
All right. I will stop recording.